Hello and welcome to everyone joining us for today's Drug Target Review webinar titled Advancing ADC Development, End-to-End Solutions from Design to Characterization. This webinar is brought to you in association with Eurofins Discovery. My name is Dr. Carrie Haslam, editor at Drug Target Review, and I'll be moderating today's session. I'm delighted to be joined by Dr. Franziska Niethling, Head of Biotherapeutic Services at Eurofins Discovery, to explore the comprehensive process of ADC development, covering payload linker design, optimization, and the characterization of ADC molecules. This webinar will also cover how ADCs are advancing in the drug discovery landscape and how ADCs can reduce damage to healthy tissue, leading to improved patient experience and long-term outcomes. Following the presentation, we'll move on to our Q&A session, so please feel free to submit your questions at any time during the webinar via the questions panel on the right-hand menu. And with that, I'll hand over to Franziska to begin the webinar. So over to you.
Thank you for that kind introduction. I would like to share with you today our view on advancing ADC development, end-to-end solutions from design to characterization. We at Eurofins work on many aspects of large molecule development. One of the ones we focused on recently is antibody drug conjugates. What I'm showing here is a very nice summary of the general mechanism of action, and you can see the reference at the bottom right where we found it. And in general, when the ADCs are administered, they circulate in the blood. You have binding of the antibody to the antigen. You get internalization of the molecule, and then it gets metabolized within the cell, and the drug is released, and you then have mechanism of the drug as intended. That's a very high level overview. There's a lot in the literature that is available. That is just a quick idea of what it is we're talking about today. In general, an antibody is conjugated with a linker to a drug. There is usually, it can be a monoclonal antibody, it can be a bispecific, it can have different binding capabilities. There could be a triggering system. There definitely is always a payload, which could be a cytotoxin or could have some other function. The linker is often, it can be a common linker. The design of the linker has a large role in whether the drug will work or not. And then you always have a conjugation site for the drug linker conjugate to the antibody. ADCs have been around for some time. There's a lot of them that are in development at the moment. The first one that was approved for clinical use was done so in 2000. This is just a very brief summary of many, many molecules that are available or being tested. So in 2000, the drug Mylotarg was approved. It's for AML, and the target is CD33. You'll see that some of the payloads are common between different drugs, as in Besponsa, which is in the fourth line, which has the same payload. And this one is a CD22 target and is used for B-cell acute lymphoblastic leukemia. There are several versions of trastuzumab that are on the market with different drug linker conjugates. In 2013, we had Kadcyla approved, which is for breast cancer because the target is HER2. And in 2021, for instance, there was the trastuzumab deruxtecan combination that was approved, again, HER2 target, and in this case, it can be used in multiple solid tumors. The key for this is that HER2 positive, HER2 expression can be low, can be medium, can be high. And in this case, it has been approved for use in different cancers, not just breast cancer. And this drug has the benefit of being able to have a high drug to antibody ratio, and it also causes a strong bystander effect. Eurofins Discovery has the capability of synthesizing drugs and the payload and linker combinations. We can do the payload synthesis. We have the capability of handling standard cytotoxins. We have the capabilities of handling very toxic payloads. We can do the combination of the linker and the payload. The linker can be either something that is commonly used or it can be bespoke. It can be specially designed. And then we can do the final drug linker conjugate isolation purification. As I mentioned earlier, commonly in the past, we have had cytotoxic payloads, but there are others that are available. I'm just mentioning one possibility here, which could be a PROTAC, which is proteolysis targeting chimeras. We can handle the different fragments that are used, do again the linker introduction and synthesize and purify the final degrader system and isolation. This is just a brief overview of what we can do. There are very, very capable chemists on site, and there is a lot that we are capable of doing. Conjugation of a drug linker conjugate to the antibody, you can do a stochastic conjugation whereby you just have an almost opportunistic conjugation of the linker and drug to the antibody. You reduce the antibody, you mix it together with the drug linker combination, and you get binding of the antibody to the drug. And you can determine the drug to antibody ratio. In this case, you would probably get heterogeneous ADCs with different numbers of drugs conjugated to the antibody. And you can stabilize these conjugates by various methods, one of them being hydrolysis. Another method of doing conjugation of drug linker conjugates to antibody is by site selective rebridging, where you do a reduction of the molecule. You aim for specific conjugation of the antibody to particular sites, and then you do the reformulation or refolding of the antibody. And again, you have potentially a better control of the drug to antibody ratio. You get increased homogeneity because you don't get as many variables in the system. And it would enable you to do more than one payload if you needed to conjugate it to the molecule. Basic antibody drug conjugate generation follows usually the same steps. You start, once you've done the conjugation, you start with a drug linker combination. You have the antibody in there. You do the bioconjugation by whatever means. There are different methods by which you can do this. You then have a mixture. You have unconjugated antibody. You have antibodies that have several different levels of drug connected, and you could have unconjugated drug in the mixture as well. You apply this to a size exclusion column, and you separate out the different fractions, so you can get a somewhat pure population of the antibody drug conjugate that you have now made. That fraction of that purified fraction of ADC now goes to mass spec, and you can analyze the drug antibody ratio. You can see what you have. You can see whether your molecule is intact and whether it is the right size that you expect. Characterization of antibody is what the first kind of characterization you want to do. You want to make sure that you haven't changed what your antibody is supposed to do. Most basic methods of doing this are flow cytometry, where you look and make sure that it still binds to your target, which is expressed on the cell surface. In this case, it's just an example of 3 breast cancer cell lines. One on the left, MDA MB231, has low or no expression very low expression of HER2. MCF7 has moderate expression of HER2, and then BT474 is known to have high expression of the target. And that shows that you haven't changed the intrinsic binding capability of your antibody. You can also do this by SPR, where you bind your target of interest to a biosensor. You just demonstrate whether your antibody still binds to that target or not. And then you can also do it by a very basic method, or probably the oldest method available, which is ELISA, where you bind the target to the plate, and you measure that you demonstrate that your drug is still binding to that target. We took this very basic concept and we performed 2 case studies. In the first case, we selected a non-toxic fluorescent fluorescein cadaverine as the payload. This would allow us to demonstrate where the drug linker conjugate is going. And we selected 2 linkers, a non-cleavable linker and a protease cleavable linker. You may recall in the very quick overview on the first slide, I showed that some of the drugs can be cleaved, some of them are not. And in this case, we had the option of putting both, conjugating both to the fluorescein. The non-cleavable linker, I won't go into the chemistry, but the non-cleavable linker and the cleavable linker were made in a similar manner. They had a similar molecular weight and they were purified. And we used them then to conjugate to the antibody. We used the anti-HER2 trastuzumab, which is a very well-studied antibody. There's a lot in the literature, and that helped us to be able to demonstrate that the ADC is doing what it should be doing. We followed some of what was in the literature. Purification, as I mentioned earlier, the first step is size exclusion chromatography. You can see that with both the non-cleavable and the cleavable linker, we got a very nice clean peak. We isolated that peak, and then that fraction is what we used in each case to characterize, to make sure that to demonstrate that we hadn't modified the antibody at the same time. In flow cytometry studies, in this case, just demonstrating the cleavable linker payload molecule, we did binding assessment by flow cytometry. As I mentioned earlier, we used MDA MB231, which has no very low expression of HER2, BT474, which has medium expression, and then HCC 1954, which has a very high expression of HER2. In the top row, we have unstained cells. And you can see in the live gate, there's very little staining in all 3 of the cell lines, whereas when we applied the ADC, very low staining, very little staining of the MDA MB231. We had moderate staining of the 474, and then we had a large number of cells that were stained in the HCC 1954. Just to put those numbers into a more visual representation, you can see that for the unstained cells, all 3 cell lines had virtually no binding to the antibody. And in the stained cells, we had very low, 2% of cells were positive for MDA MB231. 59, call it 60% of the cells were positive for the BT474 cell line. And then for the high expressing HCC cell line, we had 92% of the cells that were positive. So this binding clearly demonstrates that the specificity of the antibody has not been affected by the conjugation to the drug linker conjugate. The most basic characteristic of the antibody is preserved. Looking at internalization of this ADC with a cleavable linker, we conjugated the antibodies to a pH-sensitive dye, a Frodo dye, which is red, and you can see at the bottom how the intensity of the fluorescein increases at low pH. So as the molecule is internalized, the fluorescence increases, and you can clearly show that cells have taken up the antibody, or that the antibody has been internalized into the cells or not. In the top row on the left, we have the highly positive HCC 1954 cell line. Before means about 5 minutes after adding the antibody, just having enough time for us to process the plates and put them on the reader. This was analyzed on a Citation 5 reader. And you can see after 24 hours, there are many, many cells that are highly fluorescent, indicating that the antibody has been internalized. And then on the bottom right is the MDA MB231 cell line, which is negative. And you can see the before and the after are pretty much identical, demonstrating that, first of all, this binding top left to the HER2 positive cell line is specific, and that there is no non-specific internalization of the cells into internalization of the ADC into a HER2 negative cell line. HER2 negative cell line. With the HER2 positive cell line, we had 92% of the cells that were positive. So this binding clearly demonstrates that the specificity of the antibody has not been affected by the conjugation to the drug linker conjugate. The most basic characteristic of the antibody is preserved. Looking at internalization of this ADC with a cleavable linker, we conjugated the antibodies to a pH-sensitive dye, a Frodo dye, which is red, and you can see at the bottom how the intensity of the fluorescein increases at low pH. So as the molecule is internalized, the fluorescence increases, and you can clearly show that cells have taken up the antibody, or that the antibody has been internalized into the drug linker conjugate. The most basic characteristic of the antibody is preserved. We then take the molecule, as I mentioned, and we do an assessment of the drug to antibody ratio, referred to as DAR. You need to know how much of the drug you have conjugated to the antibody. Why? Because you might want to be able to control for cytotoxicity. You might not want to go in and blast, potentially have off-target binding or off-target effects. You can deliver it very specifically to tumor cells, and you can, to a certain extent, control the cytotoxicity by controlling the drug-antibody ratio. You must be able to quantify it, because for the same reason, so that if you need a specific drug antibody ratio, you can demonstrate that you have it. This is typically done through mass spec analysis. And you may want to control the DAR because, if you want to control it, modifications can be done to both the antibody and the drug linker conjugate. This particular, the linkage of the drug to the antibody, the conjugation, you may want to optimize it. You may need to change it. You may need to, if you find that the antibody is being affected by the number of molecules, or in vitro testing, you're finding that the DAR that you have is not perfect, you might want to optimize it. It might be that you want to use it in certain circumstances in a solid tumor, you might want to use it in a liquid tumor, could be for different applications of this molecule. In this slide is an illustration of the mass spec analysis of this molecule. In the top trace, you have the unlabeled trastuzumab. It should not contain any of the drug that you expect, the drug linker conjugate. In the middle trace, you have the antibody with the cleavable linker payload. You can see that it is positive there. There is a signal. And then in the bottom, you have something that was conjugated to a different, you have the antibody that was conjugated to a different molecule. So you don't see the expected size peak in this bottom trace. And at the bottom is just an example of the intensity of the measurement in this. And with this analysis, we calculated that the drug antibody ratio is 2. There are 2 drugs on each antibody. And we also found that there was a DAR of 4, which is not illustrated. And here on this slide, you can see that there's a DAR of 6, which is indicated by the higher molecular weight, the bigger size, the larger size of the molecule. This can be done very, very accurately with mass spec. And the team here on site does quite a lot of mass spec analysis, so they're very good at this. For the second case study, we used a linker and drug combination that has been used. It's 2 of the combinations that were mentioned in the first slide of drugs that are on the market. One is we conjugated trastuzumab with an MMAE linker conjugate, and this leads to cell death in cancer cells by a very specific method. We also used the deruxtecan DLC, and this is a topoisomerase 1 inhibitor which interferes with the replication of DNA and eventually leads to cell cycle arrest and apoptosis in cancer cells. We did this so that we didn't just do fluorescence demonstration of the characteristics of the antibody. We actually wanted to show that this antibody can lead to cell death as well. So apart from the assessment that we did with the previous antibody drug molecule, we also went all the way through to cytotoxicity assessment. In this case, we applied the ADC to both ADCs to HER2 positive cell lines. Illustrated here on the left, you have SKBR3, which has medium expression of HER2. And on the right, you have the highly HER2 positive HCC 1954. You can see that as the drug titrates out, the green line is trastuzumab alone on each of the graphs. The blue line is the trastuzumab deruxtecan combination. And then the red line is the trastuzumab MMAE ADC. And you can clearly see on both cell lines that the MMAE is a more potent toxin than the deruxtecan, and no killing, little killing with the antibody as the antibody titrates out. These ADCs, these data demonstrate that the ADCs are toxic to both HER2 positive cell lines. You can see that again that the MMAE is a more potent ADC, has a higher IC50 or a better IC50 than the deruxtecan compared to the trastuzumab. And then by the same token, checking on the toxicity of the two ADCs on MDA MB231, which we very well know by now doesn't express HER2. You can see that there's no cytotoxicity across the titration, the full titration of the antibodies. And as you saw in the previous slide, ADCs are only toxic to the HER2 positive cells, but not to HER2 negative cells. So what we've demonstrated in these studies is that cleavable and non-cleavable payload linkers that we synthesized and conjugated to trastuzumab, we were able to produce highly specific and functional and ADCs. Flow cytometry and internalization confirmed the antigen specificity, that we didn't change the intrinsic characteristics of the antibody. And we demonstrated progressive endocytosis of the fluorescein conjugated molecules, and the cytotoxicity assays demonstrated the HER2 specific action of the two toxic ADCs, as seen with the corresponding nanomolar IC50s on the two HER2 positive cell lines. So how do we go about the creation of antibody drug conjugates at Eurofins Discovery? We have quite a few requests for both doing the antibody discovery. You then have to do the characterization, and then once you've identified the right kind of antibody, you do the ADC characterization creation, and then you do the characterization again. So we can cover the full spectrum of antibody discovery, from early discovery, from target validation. We are often asked to generate the reagents, to monitor the reagents. We do protein production. You then go through the full spectrum of antibody discovery, from immunization to basic recovery, to secondary screening, to optimization by humanization, liability removal. You go through several rounds of recombinant expression. We do the manufacturability assessment both before and after conjugation of the ADC, because again, you want to make sure that you maintain the characteristics of your antibody. We also do safety assessment by various means, depending on the function of the antibody, the application of the antibody, and we can also do phenotypic biology with many, many assays that are available, and those are standard catalog offerings. Once we get to preclinical testing, we are often asked to do bioanalytical testing. These can be applied in PK/PD studies. We have been asked in the past to do immunogenicity assessment of both the antibody, the drug, and the ADC together. And we can assist in biomarker development. Once it comes to the later development of a molecule, we can build, and we have built, several lot release assays for clients, which we can then hand over to a GXP facility. We are able to make cell lines. We are able to do cell banking. We can test the formulation and optimize the formulation of molecules. And we have also been asked to build assays for QC testing and lot release. The general, as we mentioned before, the general antibody drug conjugation characterization assays most commonly are cytotoxicity. We can do this by various means. We showed you one method earlier in the presentation. We are agnostic as to the platform. We can work with whatever the client asks us. We've been asked to use commercially available tumor cell lines, such as the ATCC cell lines. We have DiscoverX target-specific killer cell lines. We can use primary cells and specific effector cells, if necessary, in combination with the specific drug as needed. As to the actual assays, we can use whatever is available in the market. We have some clients that are very fond of LDH assays. We can do cell-tited glow, and then we can do live dead assessments by flow cytometry. We can also do subpopulation phenotyping by flow cytometry to assess the population that may be affected by the drug. And then, as you saw earlier as well, we do have the capability for high content imaging. Some people prefer flow cytometry, some people prefer the high content imaging to be able to visualize their data. We can then move on to the manufacturability and stability assessment of the molecule, looking at aggregation or cleavage, see whether it falls apart. And this can be done by different methods. We have clients who present us with their protocols that they want us to replicate. We can make suggestions for protocols. We can work with clients in whichever manner they want. We can look at the buffer formulation. We can look at different buffers of the composition of buffers, different pH. We can assess the stability of molecules in that. And then we can also perform force degradation. Again, many clients have their own protocols that they want us to use, or we do we can propose the different conditions that might be useful to assess the stability of the molecule. And as also mentioned before, we can look at the affinity of the antibody by SPR, make sure that that nothing has changed during the conjugation process, and we can assess the function by various methods depending on what the application is. At the same time as looking at the general characterization of the molecule, we can look for unexpected effects. We can assess the immunogenicity and off-target binding and many other assays. Some of the immunogenicity assessment that we have been asked to do is the MAPS, MHC-associated peptide proteomics, where you actually apply the antibody, the ADC, to dendritic cells. You lyse them. You do a pull down of the HLA molecules with the peptide. You isolate the peptide. You elute the peptide from the MHC complex, and you analyze that peptide population by mass spec. Perhaps quicker processes to assess immunogenicity are cytokine storm, where you incubate your molecule, your drug candidate with PBMCs, and you look for cytokine release. The number of cytokines we assess can be tailored to your needs. A basic cytokine panel is, an average cytokine panel is about 10 molecules. We can go up from there, or we can go lower from there. We can tailor it to your needs. Another method of assessing immunogenicity is looking at T cell activation. Same thing, you use either PBMCs or a purified pan T cell population, and you look at the, you can assess the T cells by flow cytometry, looking at various markers. Again, we can go in depth looking at many markers, or you can go just to a basic assessment of 3 or 4 cell surface markers and assess those by flow cytometry. Data packages are delivered at the conclusion of the project. All the raw data belongs to the clients, and we are very good about communicating with clients. We keep you up to date on your projects, and we are always open to communication. So obviously there are many people who were involved in the generation of these data, and I'm very grateful to all of them for allowing me to present this to all of you. And I hope that you have enjoyed this presentation, and we would love to take questions.
Thank you, Francisca, for that fantastic presentation. Now let's move on to the final part of today's webinar, which is the Q&A session. So a quick reminder to our audience, you can still submit your questions using the questions panel in the menu on the right. And joining us in today's Q&A is Guzman Sanchez, Product Manager and Senior Scientist at Eurofins Villa Pharma Research. So the first question we've got here is, what do you think are the most important things to consider when designing a linker? Francisco, I think
Okay.
you might be on mute. There we go.
Yeah. I'm going to consult Guzman on this one.
Yeah, well, there are many factors to consider. So first of all, you need to know which mechanism of action are you pursuing for your release of the drug, right? Then you will also consider the attachment point to the payload, and that will depend on also on which payload are you going to use. It's not the same some auristatins or some other bespoke payloads. Also, you need to be aware of the lipophilicity of the whole system. So you might need to add branching points, solubilizing groups, etc. And also, you also want to consider which kind of conjugation do you like to your antibody? Because if you are pursuing more homogeneous ADCs, you might want to go to site-specific or even to glycan conjugations as well, not just stochastic, because stochastic is difficult to control, overall speaking. So as you can see, there are many factors to optimize when designing a proper linker. And even though you do this comprehensively from the outset, nothing guarantees that you are going to have success in the first run, right? So you might need to be doing different optimization runs just to get the best linker for your system, and that will depend also on the payload as well. So yeah, many things to consider all at once.
Oh, that's great. Thank you, Guzman. Really appreciate that answer. So the next question is, how do you optimize drug antibody ratio?
So thank you for that question. We've answered some of it in the presentation. The other part is that you could engineer the antibody to have a specific conjugation site, but you have to be careful about how you do that so that you don't change the intrinsic shape or the characteristics of the antibody. And you can also adjust the linker of the molecule either by shape or conformation. Length or the conformation of the linker to make it more amenable to conjugation to the drug.
That's great. Thank you, Francisca. So the next question is SPR the best way to select the most promising ADC candidates?
The answer to that is not straightforward. It could be a quick way to demonstrate that your antibody still binds with the same affinity that it demonstrated before you made the combination. But there are other methods of doing that. If your target is cell surface bound, then obviously you can't use SPR or not commonly use SPR to be able to measure that. So you have to take into consideration what the function of your antibody is, where your target is located, and then make the decision of how you're going to characterize that antibody, that ADC.
Perfect. Thank you. This next question is, for ADCs, what assays do the regulatory agencies typically ask for?
I think the most important question that anybody can ask and that the regulatory agencies certainly are interested in is safety. Are you going to have off-target effects? What is the overall effect of that antibody going to be systemically, not just at the site of the tumor or whatever it is you're trying to treat? They want to make sure that your molecule is stable, that you actually have a drug, that it can be stored for a while, that it's not going to fall apart. And they want to know what the toxicity of the drug, the payload is if the drug falls apart and the payload is excreted from the body. Those are the very basic questions. There's quite a lot. There is fairly new guidance available from last year that the FDA issued, and that would be a great starting point for anybody to look at.
That's great. Thank you. This question is, what are the best methods to determine toxicity of an ADC?
I think the most basic method is going to be to see whether it has, first of all, if the function of it is toxicity, you want to make sure that it kills the cells that it's intended to kill. But also assessing the toxicity can be twofold. Do you want it to be toxic? Yes, then you do what I just mentioned. If you want to make sure that your molecule is not having an unintended toxicity effect, then you have to test related antigens, you have to test related systems and make sure that there's not unintended toxicity. So we mentioned some of the toxicity, some of the wanted effect toxicity assays in that, I think it was the second last slide, and that's a good starting point.
That's great, thank you. Now this next question is very interesting, and it's what are the most common reasons for ADCs to fail clinical trials?
It's usually one of two. Either it works too well and you have unexpected toxicity that could lead to very bad comorbidity of patients, or it doesn't work well enough. So the characterization is key. You want to sort of get just the right effect of the antibody. You want to make sure that it's not so toxic that you're killing off everything around the tumor as well. And that, of course, is part of making sure that your antibody is, or that your drug is delivered very specifically to the site of where you want it to be working. And obviously, if your drug does not work, if it's not effective enough, there's no point in treating the patient because you're not going to get the effect that you require.
Perfect, thank you. So we've got another question for you, Francisca, and it's, is size distribution a challenge in ADC and for DARs?
It absolutely can be. That is where the purification of your drug comes in. It's where the conjugation of your drug comes in. It depends on, that's where the design of the linker is very important as well. You want to have as homogeneous a drug product as you can, because first of all, you want to make sure that what you're treating with is a particular drug combination, a particular DAR ratio, and that you're not just having unconjugated or a mixture of drug molecules in your drug preparation.
Perfect, thank you. So we've got time for one more question, and this one again is for you, Francisca, and it's how long does it take in your hands to have full antibody discovery and characterization followed by ADC production?
Unfortunately, I have to preface the answer by it depends. It depends on what the target is. So some targets are easier to make, and not easier, are simpler to make antibodies to. If it is a cell surface protein, you can usually make an antibody in it could be 6 to 9 to 12 months. It depends on how immunogenic the antigen, the protein of interest is. There are so many factors that go into it. You can be designing the linker and the drug at the same time, the linker drug conjugate, while you are doing antibody discovery, if you have an idea of what you want. You also have to do a full characterization of the antibody alone so that you know exactly what it is you're working with. And then once you put the two together, as I said, you do the conjugation. You have to go through quite a bit of the characterization again to make sure that the antibody still maintains the characteristics that you selected it for, that you can actually turn it into a drug, that it will stay stable, that it will remain from the beginning through its shelf life to application to the patient. I'm sorry, there's no straightforward answer that I can give you. You know, if it's a more complicated target, it's obviously going to take a lot longer to get. It could take a lot longer to get an antibody that has the functionality that you're looking for. And I think I'll wrap it up there because I could keep going all day.
Perfect, thank you. Well, sadly, that's all we've got time for today. So a huge thank you to our expert today, Francisca, for a fantastic presentation and discussion, and also thank you to Guzman for joining us. And thank you to everyone joining us online and for submitting your questions. As you leave the webinar, a short survey will appear on your screen asking you to rate your experience. If you're unable to complete it now, it will be sent to you via email shortly, and we would greatly appreciate your feedback. And for any questions that we haven't managed to answer today, please do be assured that we'll follow up with each of you individually so your questions won't be missed. So on behalf of Drug Target Review and Eurofins Discovery, thank you all for attending, and we hope to see you again soon. Goodbye and thank you for joining us.